Hi, everybody. Welcome to CF 101, Decoding CF Genetics to Transform Treatment here at ResearchCon. My name is James. I'm really excited that you guys are all joining with us to learn a little bit more about CF. And like you probably saw earlier, I'm one of the co-chairs of this event. I also happen to work in genetics research. So I was really excited to be moderating the session this evening, and I will be introducing our other two speakers in just a moment here. Before we get started with the session, I want to go over a couple of tech items related to how we can all interact. We will be taking audience questions during the session, and I'm going to work as many of them in throughout our discussion as I can. So if you have a question that you would like to ask, please submit that by looking for the icon on the right side of the screen labeled Q&A. You can also see other questions there and give a thumbs up if you want someone else's question to be answered. So when you're thinking about your question, please try and formulate that in a way that will be helpful to everyone here. We're not going to be able to answer questions about individual diagnoses or treatment plans, and we encourage you to use the information that you learned today to have a conversation with your own care team. But for questions that are not about somebody's individual medical information, we will do our best to answer as many as we can. You'll also notice a chat bubble on the right side of your screen. This is the group chat for everyone who's in the session. You can open that or hide that as you like by clicking on the icon. And if you do have a question for us, we would recommend that you use the Q&A icon to submit that, because that's easier for us to find as our session goes on. And if you have any technology problems during this panel, please go to the Tech and Program Support Room on the home screen of the event. So that's the interface that shows the different buildings. So the tech and program support looks like a lab. So I'm really excited to share this session with you, because I really hope that sharing my own experience learning about how CF works will help to give you all some additional tools to use on your journey with CF, whether you are a patient yourself and you're interacting with your own care team, or you are a parent or a caregiver, interacting with a care team on behalf of someone else, or whether you're, a friend or another loved one, or somebody that's in the support system of somebody with CF. So when I was a kid, learning about CF really helped me kind of nurture my own love of science. And that led to me going to college and deciding to study chemistry and math and biology. And it really led to this great positive feedback loop where I could use my own personal experiences with CF and my medical care to do a better job learning about science and biology. And then I could use what I learned in school, or just because I'm a huge nerd, and then apply that to my own life to help me navigate my own care and my own journey with CF better. I also really wanted to be part of this session in particular because my professional career relates to this really well also. I work at a place called the Hudson Alpha Institute for Biology, which is where I live in Huntsville, Alabama, where I am a computational biologist. And that is, I write software and manage data that helps our lab diagnose rare pediatric genetic diseases. We don't work with CF all the time, but it has taught me how important that key piece of knowledge is, that how genes cause CF. And that's really a key to all of the treatments that we have available. So I really wanted to be involved in bringing this information to other families, and that's why I really wanted to be involved in helping us all get a better understanding of what's going on. So I get to think a lot about some of the things that we'll discuss today. And with that, I would like to welcome our two other panelists. First is Eleanor Schwind, who is a genetic counselor. And Eleanor, please tell us a little bit about yourself.
Hi, thanks so much, James. So for nearly 25 years, I've been a part of a really wonderful CF team caring for children and adults with CF and their families. So I'd like to acknowledge my colleagues at Mount Sinai Beth Israel and my colleagues throughout New York City as they are battling on the front lines of the COVID-19 pandemic. But in my role as a genetic counselor, I educate and support and answer questions about CF genetics at many different points in my patients' lives. And before we get started, I just wanted to kind of bring to the forefront that a key principle of genetic counseling is really a deep respect for each person's unique ability to make informed decisions about genetic information and testing, some of the things I'll be talking about today, that's consistent with their own values and goals. And I'm just so honored and happy to be invited to participate in research.
Great, thank you so much for joining us. And now I'm going to introduce Marty Solomon, who is a CF doctor and, in fact, a member of my own care team. So thanks for being with us, Marty.
Yeah, thanks, James. So I've been involved with CF care for a few years now and started really when I was a resident physician. I'm in internal medicine, so I'm an adult pulmonologist, and I'm now currently our adult CF center clinic director at UAB in Birmingham, and I also am a clinical and translational researcher studying treatments for CF, specifically some of the therapies we'll be discussing tonight and some of the concepts. And most apropos to this is I actually direct the UAB Center for Cftr Detection, which is a center which is sponsored by the CF Foundation to help us investigate methods of looking at CFTR protein function in humans and using that for both diagnosis and as importantly, the monitoring of new therapies for CF. So very pleased to be with you guys tonight, and thanks for the opportunity to be here.
Great. So I'm going to talk really quickly about kind of how our session is going to look today. We're going to structure this as a little bit of a quick talk by each of our presenters, and then a little question and answer period. Like I mentioned, I'm going to work your questions and answers in throughout our planned discussion. So please go ahead and start asking questions and ask lots of questions. So what are we going to talk about today? We really wanted to start at the very beginning. CF is a genetic disease, so we're going to start with what that means and learning about genes, what genes are and how they lead to CF in our bodies. Then we'll talk about proteins, which are going to link those genes to those actual physical effects in our bodies. And we'll also stop to talk a little bit about the ins and outs of genetic testing for CF and finish up talking about current research. So what the whole field is still learning about these topics of how CF works on the molecular level. If you're wondering what you need to know to get the most out of the session, we've really structured this session for everybody. So we want there to be something for everyone, and we're trying not to make any assumptions about what people already know about CF or the biology. So it's great for people that are brand new to CF that will be able to learn a lot. So whether you have a loved one that just got a diagnosis that you heard about yesterday, or you're somebody like me that's been living with CF for your whole life and you just want to learn a little bit more. How you can participate is certainly the questions and answers, but we've also got some questions that we're going to launch through the polling feature. So we can go ahead and launch the first polling question now, and you will see a little bar chart looking icon that is for polls, and we're going to use these questions to kind of be a starting point for our discussion, and we want to get an idea of what you guys know about the topic already or are excited to learn. So we have our first polling question, which is, how would you describe your understanding of the genetics of CF? So if you want to click on the polls icon and answer, there we are expecting. Maybe some people will say, hey, I'm overwhelmed, I'm new to this. Maybe they'll say, I'm a beginner with CF genetics. Or maybe I know the basics, I could explain it to my neighbor. Or I have a good working knowledge of CF genetics, but I'm ready to learn more. So I'm going to go ahead and see if we have any results from this, and things are coming in. So there are definitely a few folks that are feeling overwhelmed and are very new to this. A few people say that they're beginners or that they have a pretty basic idea, and I think everyone is excited to learn more. So now let's go to our next question that will introduce some of the ideas that we're talking about. Give our tech folks just a second to get this one pushed out there to us. So number 2 is going to be the first thing we're talking about, and it is, what is a gene? Looks like we're starting to get results come in. Few people are saying that it is all of the above, or is it a molecular blueprint for parts of how our body should work? Is it the way that traits are inherited from parents to children, or is it a piece of DNA found in your cells? So we're going to start off. We've got one more. So I will say I'm really happy to see our results so far that says all of the above. That is going to be a great intro for what Eleanor is going to talk to us about. And let's go with polling question number 3. So the way I'm going to do this is we're going to start talking to Eleanor while folks are checking out that third polling question. So, Eleanor, the question here for you is, what is the genetic story of CF? And I'm going to share my screen so that we can see your slides.
Great. Well, thank you, James, and welcome, everybody. So this first diagram really sets the stage by showing that bodies are made up of trillions of cells, and almost every cell in the body contains that body's full genetic instruction manual called the genome, or the genetic code. And the genetic code is organized onto 23 pairs of structures called chromosomes. We inherit our chromosomes and the genes on them from each of our parents. One chromosome comes from mom's egg, and the other half of the pair is from dad's sperm. And each chromosome contains hundreds or even thousands of different genes, about 21,000 in total. And each gene tells the body to do something. Our genetic instructions direct how a body grows, develops, functions, and determines physical features like hair color or eye color, or whether or not you have a cleft chin. And the sequence or order of 3 billion DNA letters, A, T, C, and G, spell out the genetic instructions to make proteins in a way that cells can follow them step by step. Next slide, please. One of the genes, the one that we're here to talk about today, is Cftr, the cystic fibrosis transmembrane conductance regulator gene found on the 7th chromosome pair in this magnified photograph of the chromosomes of one cell. And everyone has 2 copies of the Cftr gene, which is made up of about 180,000 of those A's, T's, C's, and G's. The order of those chemical letters, which are read 3 at a time, instructs the cell to assemble about 1,500 building blocks of the Cftr protein. So the Cftr genetic instructions are followed in the cells of the lung, the pancreas, the skin, and other organs to manufacture gates, which are then packaged, delivered, and installed at the top of the cell. And these gates allow a part of salt molecules called the chloride ion to pass through. Spelling differences in the genetic instructions are called variants, often referred to as mutations. For example, maybe an A is found where a C usually is, or maybe a few or many letters are missing. And these genetic typos can affect if and how the Cftr gates are manufactured, or how many gates are installed, or how those gates open and close. Over 2,000 different misspellings have been identified in the Cftr gene. And not all Cftr variants lead to the exact same symptoms and severity of CF. researchers are working to figure out how many CFTR variants there are and how many really are CF causing. Some people tuning in today might have a child who had a positive CF newborn screening result, but their diagnosis is uncertain because we don't know enough about one or both of the variants identified. Some people have 2 kids with CF whose symptoms are very similar to each other, or perhaps an adult with CF and their sibling CF experiences have been very different. And even when we think about people with 2 copies of the most common CFTR variant called F508del, some may be our oldest and healthiest patients, while others with that same Delta F508, Delta F508 genotype may have serious health issues at a young age. And this is because, in part, many factors like other genes, those other 20,999 genes we talked about, the environment and care contribute to the overall physical effects, or what we call the phenotype of cystic fibrosis. And there is a database called Cftr2, which I believe is in our resources section, that summarizes what is known about many of the CFTR variants, and it's available to both healthcare professionals and the public. And Dr. Marty Solomon will give more details about different types of CFTR variants and what that means for CF treatments in a few minutes. But first, a little bit more about the inheritance of CF. So when a person has a spelling difference in only one of their 2 copies of the CFTR gene, but their second copy contains no misspellings and therefore makes the CFTR protein, that person is known as a CF carrier. CF carriers do not have CF and are not expected to have symptoms. When both copies of a person's CFTR gene contain misspellings, there are no working copies of the CFTR gene, and that's what results in cystic fibrosis. This inheritance pattern of the CFTR gene and its variants is called recessive. And if you learned about recessive inheritance in high school biology, you probably had to draw a diagram called a Punnett square. Looks like this, where big Y and little y is the color of a pea plant, and green is recessive to yellow. But we're really here to talk about people today. So the recessive inheritance pattern looks like the next slide, where mom and dad each have one working copy of the CFTR gene, represented by a check, and one that's been altered by a CF-causing variant, represented by an X. Their children will inherit the working copy of CFTR, or the one with a misspelling from each parent, with a total of 4 equally likely possibilities for each child. Starting from the right, the child has inherited the non-working CFTR from each parent and has cystic fibrosis. Or a child could inherit dad's working copy and mom's variant and would be a CF carrier like mom. Or inherit mom's working copy but dad's CFTR variant and be a CF carrier like dad. The 4th possibility is that the child inherits a working CFTR from each parent, so is not a carrier and doesn't have CF. Which means this couple has a 1 in 4 or 25% chance for each child, and a 75% chance or 3 in 4 chance that their children will inherit at least one working copy of CFTR and will not have CF. The 25-75% chance remains the same each time the couple has a child together. So no matter what the previous child has inherited, similar to how a family can have all boys or all girls or some of each, they can have more than one child with CF. So when we look more closely at the 3 scenarios for the healthy siblings of a person with CF, the chances are 2 in 3 that each sibling will be a CF carrier and 1 in 3 that they will not. So 2 out of 3 is the answer to poll question number 3. In this next example, we can see that the chances for a person with CF to have a child with CF is dependent on the carrier status of his or her partner. Here, dad has CF and his partner has been found to be a CF carrier. The likelihood that their children will have CF is 50%, and there's a 50% chance each child will be a CF carrier based on which copy of mom's CFTR gene they inherit, since they will always inherit a non-working CFTR from dad. But on the next slide is the more likely scenario, that mom is not a CF carrier. The chances of being a CF carrier are influenced by a person's ethnic backgrounds and their family history. CF is found all over the world, but most commonly among Northern European Caucasians, with a frequency of about 3 to 4%. In the US, more than 96% of people are not expected to be CF carriers. So in this scenario, the children inherit a working copy from mom each time and a non-working copy from dad each time. So all of the children are expected to be CF carriers and not to have CF.
Great. Thanks for that introduction, Eleanor. So now we're going to be able to get to a couple of our questions from the audience. One of them we got to answer already, which is that if you have CF, your siblings may or may not have CF. The next one is if your spouse or partner is a CF carrier, do you recommend getting tested before you attempt to have a child?
So it's a very much an individual decision. I think families, couples make different decisions even at different points in their lives. And it's something that they need to make together. That's one of the things that genetic counselors do, is kind of help families walk through the options, and then couples can make a decision about testing that feels right for them.
So since you deal a lot with families that are learning about CF diagnoses, do you ever encounter people that are struggling with guilt about, if they've given their child this disease? What do you say to them?
So every parent will really respond in his or her own way to learning that their child has CF. And so there's a whole range of feelings that are totally normal, and feelings will ebb and flow and change over time. But you're right, a lot of parents do express those feelings of guilt. And to them, I say that everyone has differences in their DNA, and it's what makes us unique and different from each other and diverse. And we don't have a say over which of our genetic differences are inherited, but all of our genetic differences together make us who we are.
Yeah, certainly for me, I never felt like anybody chose for this to happen to me or could have prevented it. It's like all of your other traits, it's just how things happen. So speaking of inheriting different mutations, can you clarify the differences between the term variant and mutation for us?
Yep. So as if genetics wasn't confusing enough at times, geneticists have kind of changed some of the terms that we use to describe these genetic differences. The word mutation has kind of become so common in science fiction and seems to have more of like a bad connotation. So we use a more neutral term that is called a genetic variant. And that's used to describe a permanent difference in a gene. And some variants can have a big effect on the way a gene works, and those tend to be disease-causing or pathogenic, like delta F508, and others may have very minimal effects, and we call them benign variants or sometimes polymorphisms, kind of like a typo in a recipe. If you just change the number of chocolate chips you add to your cookie, that is one effect on a recipe. But if the typo in the recipe changes the baking time from, say, 20 minutes to 40 minutes, that will have a different result on the cookie.
Gotcha. So it sounds like the best way to navigate around that part of the terminology is to say, I have a CF-causing variant, to distinguish from a variant that might not cause CF. So for me, I have delta F508, which is a CF-causing variant. In fact, I have 2 copies of delta F508. So can you help us kind of understand why there might be confusion if we use a phrase like the CF gene?
Yeah,
because that's something that I hear people say a lot is, oh, I have the CF gene.
Right. So Cftr is the CF gene, and everyone has 2 copies of Cftr. So the question is, does a person's Cftr have variants, or we could even call them mutations from time to time, that would cause that gene to stop working in one or both copies? So I think when we are more accurate in the terms we use, we can have more detailed conversations with our healthcare teams and with families.
Okay, here's one that just came in from the audience that I think is a really great question, which is, are there other genes that may impact the CFTR gene? I'll let you take this one, or Marty take this one, whoever wants to.
So of the, I'll start, and then if Marty you want to jump in as well. Of the 21,000 or so genes we have, they all interact, or many of them interact with each other. And so I think the more we learn about those interactions, the more we'll be able to come up with new and innovative treatments and understand more about why some people's symptoms may be more severe and more mild than others. Marty, do you want to think of that, any more of that?
Well, I'll quickly add to that. I think that's the right answer, Eleanor, that there have been a few studies that have looked at genes that the polymorphism or variant in those genes that co-associate with CFTR gene variants that cause you to have different phenotypes of CF. So that's one interaction. We call those genetic modifiers, which means that having that gene defect or gene variant makes it modify the severity of your CF. And there's lots of those, and those exist across the spectrum of diseases that's known, even in diseases that aren't caused by problems in one gene like CF is. And so the answer to that is a broad, complicated answer, but the answer is yes. We're trying to learn more about that, because if we could learn more about that, for instance, there are other ion channel genes in the body. CFTR is not by no means the only one, and if we could learn ways to figure out who has better function than the other ones, it might help us to fix those or augment those, or vice versa, to understand who might have more severe disease because they don't have a rescue by the other ion channels to help their lung disease or other parts of their body, for instance.
That's great. Let's go ahead and launch the next polling question as a lead-in to the next part of our talk. So this polling question will be asking us a little bit about proteins. And to keep talking for a second about that modifier gene example, I think that's one of the really cool things that we're still learning about CF is how these other genes, the other 20,000 genes that we have interact with our CF diagnoses. They may not cause us to have CF like that harmful variant in CFTR does, but maybe the way that two different people's immune system functions can help explain why they might have very different courses of CF throughout their life, even if they have that same CFTR mutation. So looking at our results, go ahead, Eleanor.
I was just say studies of siblings have really given us a lot of information about what the relative contribution of genes and environments, because siblings have the same two CFTR variants, but share about 50% of their other genes in common.
Great. So our first poll question, what is a protein, comes back, and overwhelmingly, you guys all seem to know that that is a molecular machine. And then most of the rest of you say you can't wait to find out. So I believe we have another polling question here that we'll look at, but this is going to lead us into Marty's presentation, where he's going to tell us about the CFTR protein, and that's going to explain how we get from that genetic change in the CFTR gene to actually having a disease that affects airways and the rest of our body. So that one is asking us how genes serve as a blueprint for proteins. And it looks like you guys have a little familiarity with that, but for those who don't, I'm going to lead us into Marty's part of the presentation. So Marty, I'm going to ask you, what is the relationship between a gene and a protein?
James, that's a great question. It's a fundamental question, and I'll try to answer that by walking us through the next few slides in a few different ways of thinking about the process by which and help us get an understanding of how gene mutations lead to protein defects or protein function defects, and that becomes disease. So if you'll advance, okay, perfect. So we're on this slide entitled Genes Are Essential for Building Proteins for Normal Health. And so this slide introduces some key terminology in answering this question, and it will set the stage for a set of analogies that will help guide us through understanding how protein defects can at least partially be fixed with drugs which are available now for some CF patients, people with CF, to help the disease process along. And so in this slide, we're using the analogy of genes as a hard copy of a set of instructions or blueprints for the production of proteins along a very well-defined assembly process. To make many copies, the builders of proteins, known as ribosomes, you can see in the middle, the very middle diagram, they each need a portable copy of the blueprint to build genes on a larger scale along the assembly line. This copy of the gene blueprint is known as the mRNA or messenger RNA. Once the ribosome builder completes the task of assembling a protein using the blueprint it's been provided, the protein can then go to its location in cells and perform a function. In the case of the CFTR protein, that function is to act as a control gate, the concept that Eleanor mentioned to us earlier and introduced to us, which regulates the movement of ions and water at the surface of the cells, represented throughout the next few slides as a fence separating the inside from the outside of cells. So in the slides we'll review for the rest of the talk, the CFTR is a gate, and the fence is the cell membrane or the surface of the cells that separates inside from outside, where the business of CFTR protein is happening. So go to the next slide, please. On this slide, the far left diagram reinforms us that the gene blueprint leads to proteins, to the production of proteins, which are molecular machines of cells, which you guys are correctly, from the polling questions, already understood that concept. In the case of genetic diseases like CF, the blueprint defects result in protein defects and then disease. Now, if you'll turn your attention to the middle diagram, this diagram reminds us about the very important processes that happen when CFTR protein fails to work in the lungs, which is the most affected organ in people living with CF. In this circumstance, the lack of proper gating, or the gate function of the protein, leads to lack of movement of ions and water, which results in both thickened mucus and the impaired ability to kill infections like bacteria. When this happens, this sets up a destructive cycle at the bottom of the diagram of recurrent infections, inflammation, airways damage, and can result in permanent and end-stage lung disease. If you'll advance the slide once, James. And then the far right diagram reminds us that the CFTR protein has a function throughout the body. And so what we can see is that the phenotype, or the way that CF manifests as the disease process, can be present in many, many organs throughout the body. And this slide just demonstrates some of those organs in which the CFTR protein dysfunction leads to disease. So, if you'll cue up the first video, please. The video we're seeing now was produced by the CF Foundation a few years ago. There have been some updates to this video, but I think this one's a little bit clearer for the purposes of our discussion today. This video is designed to drive home an important concept. In the video, we've traveled down into the lungs, and we'll zoom in, as we can see, on the cells that line the lungs. As you can see, as we zoom in to the top of the cells, in that membrane, or what we're going to call a fence for our analogies coming up, the CFTR protein is helping to regulate the mucus and allows for easy flow of mucus across the cilia, or hair-like projections, which are moving, as you can see, back and forth to sweep mucus in and out of the cell. As we zoom back in, we'll examine the circumstance when the CFTR protein is not working. As you can see, the ions, those yellow dots, can't move in and out of the cell properly. And as a result, the mucus begins to thicken and becomes sticky, and it paralyzes the motion of those cilia. And then it traps infection and inflammation in the airways and causes damage. And thus, the CFTR protein defect results in impaired lung clearance and lung disease eventually. Now I'll turn it back over to James to tell us a bit about the CFTR protein structure and answer some questions for us.
Great. So I had a neat little object that I wanted to share with you guys quickly. This is a three-dimensional model of the CFTR protein. A colleague of mine, Jeremy Prokop, and his lab developed this and had it 3D printed. But you can see that the way that this protein is shaped really impacts its function. If you kind of look kind of in the bottom there, you might be able to see that there is kind of a channel where the ions go, and this section here in red is what sits in that membrane of the cell. So you can really see how that shape that this whole thing folds into controls how well it's able to work. And if you can see this part right here that's kind of in purple, that is the Delta F508 variant, which is the one that I have. And I think that's a great lead-in. Marty, can you start to tell us how different mutations in that protein can cause different kinds of problems? So let me phrase that a little bit differently. Can you tell us how different gene mutations lead to different problems with the protein?
James, another great question, another fundamental question in understanding both disease process and also treatments, especially in CF. If you'll advance to the next slide, please. Okay, prepare the next few slides to offer a few ways to understand the process that gene blueprints help cells to build proteins and how that relates to protein dysfunction and treatments. In this first slide, I'm summarizing the processes that blueprints are copied to message RNAs. I told you that messenger RNAs are necessary to have a lot of copies for builders of proteins to be able to rapidly produce proteins and construct them. This process is known as transcription. Then the ribosome builders manufacture proteins from these blueprint copies in a process known as translation. In the post-translation process, the protein on the assembly line is then quality checked and then packaged for shipping to its proper location in the cell. In the case of CFTR, that's the cell surface or the cell membrane. If all works well, there's adequate functioning protein to service the body, the cell, and the body's needs, and therefore that functions for health in the patient. You go to the next slide, please. This next slide will help us relate protein function to the unique aspects of the CFTR protein in CF. This slide shows several scenarios of the lack of CFTR function, which include, if you'll click through, James, a complete loss of CFTR, which we'll discuss in a few moments. If you'll keep clicking, please. Where the CFTR is not quite made in an adequate amount, and therefore it turns over in the quality control checking process. Where the CFTR protein, the gate is physically obstructed. You'll keep clicking, please. Where the CFTR itself cannot open properly. And you'll keep clicking. So in this circumstance, we can see that different patient mutations prevent normal CFTR function in various ways. Okay, so this next slide is a set of diagrams to help us all understand the types of protein problems that arise and examples of common genes that some of you or some of your family members may have that cause them. I've included some familiar symbols so you can get a sense of how the protein assembly line process, to go back to the previous slide, please. I've included a few of our common analogies so you can get a sense of how the analogy of the protein assembly process, the builder, the hard copy, the gates, and the fence relate to different mutations. The first of these types of mutations is what's called class 1 mutation. And in that case, the gene blueprint does not call for making gates at all. An example of this is the W1282X mutation. These are also called premature termination codon mutations. So anything that have an X in the name are class 1 mutations. The common gene variant or gene mutation, F508del, falls into the class 2 mutation. This mutation causes the blueprint to call for only a few faulty gates, many of which don't make it out of the assembly line quality check process, and therefore very few functioning gates reach the cell surface where they're supposed to. In contrast, class 3 and 4 mutations are mutations in which, in the case of class 3, the mutation results in a protein that has a gate, but it's locked shut permanently. An example of that, the most common of these, is the G551D mutation, which was the first mutation some of you may be aware of that's treated by Kalydeco or ivacaftor. The next is the class 4 mutations, in which the mutation causes a blocked gate. So again, we have a gate that's operative in the right portion of the cells on the surface, but the gate has a physical block to it, and it can't work. And then finally, class 5 mutations cause the blueprint to only make a few gates. In other words, we have a little bit of a decreased number of those messages or blueprints to operate from, so therefore the builder can't ramp up production of proteins on the assembly line. And in the case of CF, the appropriate amount of open gates are made. So in patients that don't have disease or a mutation that does not cause CF or a variant does not cause CF, that's the circumstance in which the appropriate amount of open gates are made. So if you'll turn to the next slide, please. Okay, so this next slide relates the processes of protein production to the eventual function of CFTR protein on the cell. CFTR modulators are drugs which have been developed to modulate, to augment the dysfunctioning protein due to the breakdowns in these processes caused by gene mutations. The next set of slides will use a fun analogy. One I did not develop, was developed by some smarter scientists than myself, but I think really helps us to understand the way in both treatments work to correct protein defects, and how a couple of the common gene mutations which have been worked on by CFTR modulators are being helped at current. To go to the next slide, please. Okay, in these next slides, we'll use a common analogy that I introduced earlier. Namely, the surface of cells is represented by a fence separating, in this case, a field into two pastures. In this analogy, our ions, or chloride ions, are cows, and they need to get through the fence to reach the greener pasture land and the brook across the fence on the other side of the pasture land. But because cows are not able to jump through fences, they did an easy passage through a gate that represents the CFTR protein moving the ions across the cell surface in this analogy. If we go to the next slide, please. So, this slide helps us understand the situation of class 3 mutations, in this case, the G551D CFTR. In mutations like the G551D, where the building process produces a gate in adequate amounts, but in this case, the gate is permanently locked, as we discussed earlier. If you'll advance the slide twice, please. As you can see, once we add a modulator called ivacaftor or Kalydeco, this drug acts as a key to the gate. And if you'll advance the slide again, this allows the gate to open, and then our cows cross over and over to the greener pasture land with the brook across the fence, much like ions would be doing in the presence of the potentiator drug with G551D mutated CFTR protein. To advance to the next slide, please. The next few slides will walk us through the development of the modulator combinations that treat the most common CFTR protein defect due to the F508del mutation. This mutation is a bit more complicated because it results in a faulty building process that causes few or no gates to be assembled in the fence. So you can see this is the fence separating the cows from the other pasture land with no gates at present. And in addition, those gates, if present in the fence, are also somewhat locked, like the G551D gates were earlier. So turning to this slide, we see that in F508 del, there's a fence, but no gates for the cows to pass through to the other side of the pasture land. If you'll advance the next slide, please. In this slide, we can see that if we add a drug that helps to stabilize the gate, what that means is that helps to add a builder who can correct the quality control issue in the protein assembly line process. In fact, if you advance the slide, this helps the ribosome builder to pump out somewhat stabilized proteins that actually do make it to the cell surface. But if you remember, I told you that the F508 del, we still have a problem with the gate opening properly. So then, in addition to this corrector drug, which corrects that unstable produced gate, we can add the drugs known as potentiators, like we did with G551D. If you'll advance the slide. And again, these drugs act as a key to open the gate. And if you'll advance the slide, this allows, yet again, our cows, when these drugs are used in combination, to reach across to the greener pasture land. And we can see that when these drugs are paired as combinations of correctors and potentiators, we have a familiar modulator drug like Trikafta, which is the combination of tezacaftor, elexacaftor, and ivacaftor, 3 drugs, 2 of which are correctors to help to stabilize the protein production process, and then the potentiator drug to act as a key to open the faulty gate opening mechanism. And so in combination, this works to help with our F508 del mutation. And thus, the Trikafta, which was approved last year, we'll discuss in a few moments, and I'll turn it back over to James.
Great. Thanks, Marty. So now we are going to work in a couple questions that have come up. What of the different classes of problems with the builder or problems with the gate, what class does a splicing mutation fall under?
Okay, so splicing mutations usually can fall under a couple different areas, but they usually fall under the class 1 mutations because oftentimes the splicing causes you to make a faulty message. So a splicing variant means that the gene is supposed to be cut into segments and assembled into the final blueprint copy, make a very efficient message copy for the ribosome builder to use to produce a protein. When that splicing is off kilter, it weaves that story together in a way that almost makes no functional blueprint copy. So usually the splicing mutations end up becoming non-functional proteins. In other words, they don't make protein that's functional at all to be used in a cell.
Great. So since we've been talking about different ways that this molecular machine, the Cftr protein, can break down, let's go to Eleanor with a related question, which is that if all of these different gene mistakes cause CF, why is it so important to know which one I have, and how can I find out?
A great question, and there's a few reasons to know which CF causing variants you have. The major reason to know what we call the Cftr genotype is to make sure that you have access to all the available treatments that Dr. Solomon just talked about, ones that can be prescribed based on your genotype, or also to know who is eligible for which research studies. Of the 2,000 different variants, Delta F508 we know is extremely common. About 85 to 90% of CF is due to having 1 or 2 copies of Delta F508. After that, there are about 22 other variants included on most, if not all, CF genetic tests offered in the United States. But after 23, the number of variants tested is not the same in all laboratories. And sometimes genetic testing doesn't pick up a person's variants. This can be true for patients or people who have symptoms and need diagnostic testing, or for carriers. So if you don't know both your CFTR variants, it may be that you need to be tested for more. One way to do that is the CF Foundation's Mutation Analysis Program, or the MAP program, which is run out of Johns Hopkins. And there are also commercial labs that perform very broad CF genetic tests, either by sequencing or by testing for hundreds of different CFTR variants. And just one other point, if you have a sibling, aunt, uncle, or cousin who wants to find out if they are a CF carrier, it's really important to make sure that your family's CFTR variants are known and that those are included on the test that's being done on your relative, because otherwise the test result may not be interpretable, especially if it's a negative test result.
So there was, there we go. A question I thought was really great, which is, why could somebody have a delayed diagnosis of CF as an adult?
Should I take that one, Eleanor, or you want to take that one?
Yeah, I'm going to chirp that to my adult CF specialist.
Sure, okay. So we make a lot of diagnoses each year. Just in our CF center alone, we probably diagnose 15 or so people with adult, we call adult or delayed diagnosis CF. And that usually is because one or more of the gene variants or mutations is not a fully defective, does not cause a fully defective protein function. So what I mean by that is there is some protein being produced, and that protein being produced oftentimes has some function as a gate, okay? So it can be variable, and that variability also, with the help of genetic modifiers and other things that can affect the person also, which are more difficult to measure, all of that can result in what we call a milder phenotype or a delayed onset phenotype, which means that as opposed to being affected with lung and pancreatic issues early in life, you may not have pancreatic issues, you may have the absence of pancreatic issues, and the lung issues may be less severe or delayed until later. And it all goes back to what we've just been talking about today. In those patients, there is some residual function of the protein because the genetic blueprint does not result in no functional protein being made at the cell surface of the cell. So it all relates back to what we've been talking about today.
And this could also be similar to what Eleanor was talking about earlier with the testing panels, where they may, the newborn screening type test or the test that you might get, you know, at your pediatrician's office as opposed to at a CF center might only be looking for those common, you know, 25 or 50 mutations. And if you have a rare mutation that was not on that list, you might need more extensive testing.
Yeah, and that'll segue very well into the last part of the discussion today about rare variants and other variants we're trying to target. So
yes, and we've got about 8 minutes left, so I'm going to start those slides.
Okay, great. Okay, so if James, should I go ahead and discuss these slides now then?
Yep.
Okay, great. So, James, in these last slides, I'm going to attempt to link the important transformative work that I've been summarizing, hopefully in a way that everyone can understand, with some perspectives on the future. Now, this is not meant to be comprehensive because there are other sessions during this ResearchCon in which there are going to be more comprehensive discussions about paths to cure and other modulators and other variants. But I wanted to link this to what we've been discussing today to bring it home. Okay, in this next slide, James, if you'll click as I proceed through a timeline, this timeline represents all people with CF as 100 dots, okay? If we group patients based on response to modulators that are available, at least in some countries, in this case, mostly in the US, although some of these are available in Europe, we can construct a timeline. So in 2012, about 5 to 10% of people were discovered to have an ivacaftor or kalydeco-responsive mutation, G551D or the like. Then in 2015, there were drugs which were developed which treated patients with 2 copies of F508del, or we call homozygotes. That's like what James' genetic situation is. Then, in the landmark discovery we just talked about earlier in 2019, we saw that we could expand the treatment of people that had only 1 copy of F508del mutations, and then any other mutation that did not result in protein functions. We call those minimal function mutations. And if you keep advancing, we found that, in fact, all of those patients, about 85 to 90% of patients, depending on where you survey patients across the world, were covered with drugs such as ivacaftor or Trikafta or similar combination therapies. But going into the future, we know that there are still patients that have hundreds of different mutations, some in less than 5 patients, for which there are currently no drugs which activate or work on to correct the CFTR protein defect caused by those genetic variants. So that brings us to a question: is how do we develop treatments and cures for all? So this next slide was designed to help clear up a common misconception from the timeline we just discussed. Namely, we attempt to explain the difference between rare mutations, which are characterized by the presence of only being present in a few patients, and may or may not respond to modulators. And we contrast that to non-responsive mutations. So non-responsive mutations are mutations that don't make protein, like we discussed about some of the splice variants or some of those premature termination codon or X mutation variants. They don't make protein. So the defect is in the assembly process of making enough of the message RNA or having an effective message RNA for the ribosome to assemble proteins by. And in this case, since little proteins being made, we can't fix that alone with a modulator, either a potentiator or a corrector. So something else will have to be done, and we're going to discuss that very briefly in the rest of this section. And as you can see, if you'll click the slide again, you can see there can be overlap between rare mutations and non-responsive mutations. So some mutations that aren't responsive to modulators may be rare and vice versa. So they're not mutually exclusive, but they are different, and they'll be important for us understanding how we approach paths to cure and treatment of other mutations in this circumstance. If you'll advance the next slide, please. Okay, so this slide returns back to our cow analogy with the field, and it shows us that the circumstance of a non-responsive mutation, using our previous analogy. In the circumstance of a non-responsive mutation, we can see that the fence has ineffective blueprints to instruct the builder to start producing protein, resulting initially in no gates in the fence. However, we can replace or edit this fence, so we can replace it with a working fence to start with, or we can replace it and maybe work on the function of the gate itself, but by new drugs. And either of those are viable circumstances, which I'll walk through in thinking about the defective CFTR gene. So this will walk us through different scenarios of how to fix defective CFTR genes in the future. One option is we correct the gene itself, and that can be done by delivering new gene. That's called gene therapy. Or we could fix the gene blueprint by editing it. So Eleanor used the concept of spelling defects in the gene blueprint. So we could correct the spelling defects directly in the blueprint and allow it then to function as a good blueprint for working the protein through the assembly line process. The other option is to see if we can further work on fixing the protein with the modulator concept. So by that, we could either deliver new or correct the faulty blueprint copy. So we could make a copy by which the ribosome could then say, forget the hard copy blueprint being defective. We can just give a new blueprint copy, which works and circumvent the problem of the hard copy being not working. Or we could fix these new mutations by other drugs. One example that is the development process of what's called read-through agents, which actually skip through the premature termination message of the translation process that happens in X mutations, which account for about 10% of all CF patients worldwide. So as you can see, there's a lot of different ways we can approach this, and if you guys are interested in this, I would encourage you to work through some of the other ResearchCon sessions which are focusing more on this, and look at the CFF website for more information about this path to cure. And so having outlined these processes in some detail, I want to close with a complementary video to the one we viewed earlier. If you'll switch over to the video and cue that up. So this video starts with the circumstance of already thickened mucus and infections in the lungs, and shows that the ultimate goal of all the concepts and treatments we've presented today in this session is aimed to restore protein function at whatever level of the building or assembly line process its production breaks down, resulting in restoring mucus clearance in the lungs, for instance, and ultimately preserving and improving health for people living with CF. And so with that, I'll say thank you very much and hope we've been helpful in the sessions that the portion of the sessions I've been discussing to bring these points home.
Thank you so much, Marty, for sharing that with us. And Blue Jeans is giving me a strange error. I hope you can still hear me. I want to thank you, Marty and Eleanor, for joining us. I wish we had more time to keep discussing because there are plenty more questions that we could have gone into. But I really hope that this has informed some of our viewers, and that it will really encourage you to learn more about that biology of CF, because I think it's cool and I think it's a huge nerd, and I really hope that this inspires you to learn about CF and have different conversations with your care team, and just use that knowledge to help you on your own journey. So thank you so much for joining us, everyone, and thank you, Marty, and thank you, Eleanor.
James, there's a couple of really good questions. Do we have time? I think we can stay on a bit longer. Is that correct? There's a couple of really good questions about the last portions of the talk, if I could answer those real quick.
Yeah, please do.
So someone asked, if you try to fix the defect by skipping over a stop codon, it's a great question. How do you avoid skipping over other important stop codons in the genome? This is a great question. And the reason for that is that the drug has to be very specific for skipping over a faulty stop codon. So there's actually some DNA or parts of the message RNA where you're actually skipping over that stop codon message that signals this is a proper stop codon or not a proper stop codon. So there's some signals in the actual DNA molecule itself, and so it can recognize the difference. And so the drugs have to be tested to recognize those proper versus improper stop codon signals when they're being done. So if they don't do that, they would be rejected as a potential drug in the development process. But that's a really good question.
And Eleanor, can you really quickly tell us how somebody can access genetic counseling if they have a family planning concern or a tricky diagnosis situation?
Yep, I've got two thoughts about that. Always go to your CF center first. Even if there's not a genetic counselor working directly in the CF clinic, I would say almost every institution that has a CF center does have a genetic counselor associated somewhere. Another option is www.nsgc.org, which is the National Society of Genetic Counselors. And then a little secret I will tell you about is that the CFTR2 website has a Q&A section, which is monitored by a wonderful genetic counselor, Karen Rary, and she will answer questions about rare mutations, give you new information, and if your question is more about family planning, she will reach out to the CF Genetic Counseling Committee community to see if someone would be willing to talk to you.
Regarding rare mutations, our center, as well as a group of centers across the U.S., are leading some studies to look at modulators. So if those are people that are interested in those, they have rare variant mutations, please feel free to reach out to us. We can see if you're reasonable, maybe a reasonable candidate for studies. Do we have time for a couple of questions?
That is so important, because I see that in my work too, that it really takes all of these rare patients getting together to be able to create that knowledge. I think we have time for maybe one more really quick one, and then I want to make sure to respect everyone's break time.
There was another question. I saw the other question I saw that was interesting was, how do you actually edit a gene and have it be edited in all the cells throughout the body? So as it turns out, there's been literally maybe some of the most pioneering work done in that field called CRISPR-Cas9. And that is a system that was borrowed from bacteria originally. They have a system that can cut out genes and put them back into cells. And they use those to edit genes in the actual living organism to do things like fixing genes and whatnot. And so we can borrow that system. It's been done. And actually in the genetic disease known as sickle cell anemia, this has already been done successfully in a study that's ongoing at the National Institute of Health. Some of you guys may have seen on CBS a few weeks ago, there was a study about a young African-American patient who has sickle cell, and she actually was treated and has now resolved her sickle cell from the CRISPR-Cas9. So this is a highly targeted editing process that uses other proteins called enzymes. They're enzymes which cut the DNA out, and then they insert using the Cas9 protein, they actually insert a new piece of DNA into the existing DNA, and it's very, very conserved, which means that once it's there, it tends to stay there. And so if you can hone it, which means get it to the right spot without damaging other parts of DNA that are important, and effectively, which is part of what we do in the lab, then you can actually effectively and permanently edit DNA. Now, that's not ready for prime time in CF, but it is being studied at the pharma level and also at the lab level right now. So it will be something we hope to realize for CF patients. So
There'll be more information on that in other ResearchCon sessions because I think the key there is that it's a lot easier to take someone's bone marrow out, do something in the lab, and put it back. But we have no idea how to make that work in the lungs or throughout the whole body yet. So that's really where the research needs to go next. And with that, I really want to give everyone time for a break. So thank you so much for joining us. Thank you for staying a few minutes over, and hope you have a great rest of your ResearchCon.
Thank you all very much.